that is found in different parts of the body. So the protein that is coded for is found in different parts of the body. So maybe found and used in different parts of the body. So a good example of this is the gene that codes for a protein called fibrillin. So this is just an example, one example. So fibrillin is a protein found in connective tissue. And we know that connective tissue is very diverse, right? It includes fat, and it includes blood, and it includes bone and cartilage. So this could cause, if you had a defect in the gene that codes for this protein, then it could cause many, many changes in the phenotype. So this example is what is referred to as Marfan's syndrome. So if we look at just a diagram or an image that kind of illustrates the different changes in the phenotypes, so you could write down that they have eye problems, right? They have abnormal chest, so it's kind of caved in because connective tissue obviously kind of makes up that hyaline cartilage, makes up the part of the ribs, their connection to the sternum. And then there's also heart and lung problems because there's connective tissue in, for example, in the aorta. And there's also really important connective tissue in the heart that makes up the valves. Okay? And then they tend to be tall and thin, and they have really long arms and legs and fingers. Okay? So it's kind of interesting to note that they believe that President Lincoln had this particular disease. And so they say that he probably would have died of heart issues, even if he had not been shot. Right? So maybe that makes it feel better. Okay.
Is it treatable nowadays?
No.
People die from.
They can treat it. I mean, they can monitor it, right? And so they could probably go in. They might be able to replace valves. They might be able to rebuild aortas. So it would be, but I mean, still, President Lincoln lived a long time relative to the average lifespan at his age, right?
I had a friend that had heart attack when she was a female. And they don't recommend they have children. Mm-hmm. And she did it.
Oh, wow.
It's very, very, very difficult.
Was her child okay?
Her child was okay.
Oh, wow.
Did not have.
Oh, interesting. Okay, another example is called porphyria. So this is another example. This isn't Marfan's syndrome, but this is called porphyria. And porphyria is a chemical that accumulates in the body because our body needs to break down hemoglobin, and part of the hemoglobin has to be broken down, and it produces porphyrins. And then porphyrins cause a accumulation in the body, and so porphyria has some other effects. So what happens is that you have a missing enzyme, and porphyrin accumulates. So this is from the breakdown of the heme in hemoglobin.
Is this in the book somewhere?
Not this diagram, no. So you just need to know that there are many different phenotypic characteristics that are associated with developing porphyrin. And so, for example, you could just use some examples here. So they have dark red urine, right? They have abdominal pain, constipation, weak limbs, stupor, convulsions, and then sometimes they go crazy. Right? And it's kind of interesting, it runs in families. And so, for example, one ancestor could have a problem with dark red urine, but none of the other characteristics. And then another ancestor could have problems with their behavior. And so there is a movie, and it's probably also a play, which is called The Madness of King George. And it's kind of a funny play, but it's kind of sad in that King George in, I'm assuming the UK, had this illness, and he occasionally would go crazy, and they would kind of have to lock him up, and they would have to kind of tell the people that he wasn't going crazy, right? And so they would lock him up, and then he would get better, and then he would go crazy again. And so this was just another thing that, another example of pleiotropy, right? That was found in kind of a famous person. So it's amazing that one gene could cause all of these different effects, right? Because also, we'll talk about this, but genes interact with the environment, right? So the environment can also play a role in like maybe which ones of these they are more likely to have problems with. Okay, so next, this is different from epistasis or pleiotropy. And so poly means many, and genic means many genes. So many genes, and so we could actually say that eye color is also polygenic in that there are multiple genes. There's not just the A, B, O, but there are many genes that regulate eye color. And so one good example of this is skin color. So there's a possible 6 different genes. Maybe I'll put 6+ because they don't really understand it, right? So polygenetic, right? Many genes affecting skin color. And we also know that this is also determined by the environment. So I'll put plus environment. Because, right, as you're exposed to UV radiation, ultraviolet radiation from the sun, your skin darkens in order to protect the genetic material from being damaged by sunlight. So that's why you tan. It's actually a protective mechanism. Okay. So we're just going to use a model example. So we're going to use a simplified example. So sometimes they call, when you have simple examples in science, they call these models, right? So we're going to talk about 3 genes, and the genes are going to be A, B, and C. And I'm going to say that these 3 genes have only 2 alternative alleles, and they are completely dominant. So when we look at the A, B, C, the dominant alleles, right? They code for melanin. The recessive alleles do not code for melanin. Okay, so they do not code for melanin. So this is just a simplified, just to demonstrate some of the properties of polygenic inheritance. Okay, so for example, we could have people of intermediate color, and they could potentially have children, right? So both of these individuals, they would be heterozygous for all 3 genes. So they would be like medium in color. If we looked at the distribution of color, this is kind of average color, right? And so if we look at how these genes behave, we could see that they could separate out if they, and the assumption here that I'm going to talk about later is that we're assuming that they're not on the same chromosome. So A, B, and C are all on different chromosomes, okay? So that's for later when we talk about linked genes. Okay, so you can see that if this individual inherited this little a and that little a, right, they could be this. If they got this little b and this little b, they could be this. If they got this little c and this little c, they could be this, right? So this would be very light skin. Very light skin, right? And then you could also have the opposite happening. So you could get big A coming together, big B coming together, big C coming together, and this would be very dark. Okay. So it's incorrect to say that the offspring are always intermediate between the two parents, because two parents of intermediate color could produce either very light offspring or very dark offspring. And this is really important to understand, given kind of racial ideas about race and skin color. So if I'll come back to that in a second. So if we look at this family, for example, these examples, these are twins. They're not identical twins, but they're fraternal twins. So remember, would that be monozygotic or dizygotic? So dizygotic. So these are two separate eggs, right? They were fertilized by two separate sperm, and then you can see that they are very different in color, but they are siblings.
And there's that one family that they've got the twins. That one's black, one's really pale and red hair.
Yeah, there's other examples of this.
Yeah.
So a few years ago in Europe they have a dark-skinned population, and these are called the Romas, and they have kind of the derogatory name of being gypsies, right? And so the gypsies or the Roma in Europe, the Romas are kind of considered to be lower, class. They have lots of prejudices against them. And so when we look at this example, this little girl, the assumption here is that this little girl must have been stolen by the gypsies, because they thought that there was no way that the Romas could have had light-skinned individuals, right? And so they were actually going into the populations and actually forcibly removing these children. And then they did the genetic test and they're like, oops, this is your child, right? In this case, these are the aunt and uncle of the child. But this child does belong to that family. And so it's really important to realize that, idea of polygenic inheritance. Okay, so if we go back to our model, one of the things that we see about polygenic inheritance is that we see continuous variation. So in this model, we see continuous.
If you continue to go through all the different combinations of genotypes for what we were just doing.
We're going to do that right now. Yeah. So that's where I'm moving. So then we have versus discrete variation. Right. So we have continuous versus discrete variation. So continuous variation means that there's not just like dark and light, right? There's a continuous shade of individuals. And this is very different from what Mendel did. And Mendel was actually very smart to pick pea plants that had discrete variation. So they were either tall or they were short. Imagine if he tried to do his experiments where the plants were like tall and a little bit taller and a little bit taller, and then short, then he wouldn't have been able to find out the things that he did. So he purposefully chose traits that were discrete, right? Versus round versus wrinkled seeds, for example. Purple versus white flowers, okay? So we see this continuous variation. So what we see in the population, okay, so this would be the number of individuals, and this would be increasing along this axis, and this would be skin color from light to dark. Okay, so what we see with this continuous variation is we see a bell-shaped normal distribution. So this is what is called a normal distribution. Okay, so what that shows me is that there are a few life individuals. Let's see, is that right? Did I screw that up? I think I screwed that up. Let's see, I think I wanted this a different, if you flip this axis, let me do this again, sorry. Okay. What I wanted was skin color on this one. Okay, and this is number of individuals. Okay, so now this makes sense. So this is light, and this is medium, and this is dark. Okay, so I had to shift my perspective there. Okay, So what you see is most of the individuals, right, are some intermediates. A few are light and a few are dark. Okay. So this diagram, I would never make you do like a Punnett square, but you could do a Punnett square for 3 different genes. And so if you look at the probability, you could do a Punnett square, and if you looked at the probability of getting little a, little b, little c, that is going to be very small, right? The probability of getting all dominant alleles is going to be very large. And then you can see that this distribution changes and becomes more common when you go to having intermediate. Yes.
So would albinism be?
Albinism is something different. So albinism is generally due to a enzyme that is necessary for the production of melanin. So this is not albinism. So this is just showing like very light-skinned people versus very dark, normal distribution. Okay. Okay. So another good example of this would be height, right? So we see if you lined everybody up, most people are of an average height. Some people are really short, some people are really tall, right? But you see a normal distribution. And height is also determined by nutrition, right? So we see, it's kind of interesting, the question, as when we're starting to see people that are becoming taller, right? So that we're kind of shifting from shorter people to, and we're actually becoming taller as populations change. And it might be due to genetics, but it also might be due to nutrition. So it kind of promotes, better nutrition kind of promotes a taller height in general. Okay, so that's polygenic. Okay. So next we're going to talk about how the chromosomes play in relationship to patterns of inheritance. And we've already looked at sex-linked inheritance. So now we're going to look at linkage, for example, in terms of autosomes, because there's obviously a lot more autosomes than there are sex chromosomes. And on each autosome, we have many, many different genes, okay? So this is what is referred to as, or can be referred to as linked genes. And these are genes that are found on the same chromosome. Now, when Mendel was doing his work on peas, he had no idea what genes were, and he didn't know that they were actually grouped onto chromosomes. So the scientist who came up with this idea was another person named Thomas Morgan. And he didn't work with pea plants, he worked with fruit flies. And this is in the 1920s. So you remember that Gregor Mendel was like 1850. So we almost have like 100 years before we figured out that genes are grouped onto chromosomes, and that is going to influence the way that they are inherited. Okay. So if we look at fruit flies are really interesting because the fruit flies that they talk about, the example that they talk about in your book is actually X-linked, and it has to do with eye color. And so fruit flies come in, you know, when you have your windows open, you have like fruit or rotting something, you know, vegetables that have gone bad. And they come in and they lay their eggs on there. And so every once in a while, you get a mutant individual coming in. And so in Thomas Morgan's research lab, they had a white-eyed individual come in, very unusual, and they were able to capture it and then look at how that gene or that mutation was inherited. Now, you can also, really interestingly, cause mutations to occur. And so fruit flies have been subjected to things that cause mutations like X-rays. So early on, they just subjected fruit flies to X-rays, and then they looked at what mutations they found, right? And then they started kind of playing with crossing individuals with different mutations. And so an example of this can be seen on this image. Okay. So this is an example of, this is kind of what is sometimes referred to as the wild type. And you don't need to understand wild type mutants, but if you were to take a 300 level genetics class, they talk about the wild types and they talk about the mutants. And a positive actually means wild type. So in this particular example, this is actually a gene that codes for wings, and in its mutant form, it causes the wings to become all shriveled up, or they're said to be vestigial. And so this is the normal gene that codes for normal wings, and this is the mutant allele that codes for the abnormal wings. So this is what the fruit flies look like when they fly into your kitchen. This is what a mutant fruit fly might look like. So they aren't actually able to fly, they can only walk around, and they have black bodies. And so all over the world, they have these labs, genetic research labs, and they have these breeds of fruit flies that they kept going for many generations, like maybe 50 or maybe even 100 years. And they are pure in that, like this one would have these mutations and only these mutations. And so if you're doing research, you could, take that strain of fruit fly versus this strain of fruit fly and bring them together and see what happens. Okay. So if we look at they have only 4 chromosomes, 4 pairs of chromosomes. So if we look at chromosome number 2, one of the things that you see here is that on chromosome number 2, there are many different genes. So there, for example, black body versus gray body, right? Purple eyes versus red eyes, long wings versus vestigial wings. So all of these traits are linked together on chromosome number 2, okay? So I'm going to show you just kind of how Morgan was able to prove that genes were linked. Oops. So we'll kind of go through that idea of linkage. Okay. Oops. Kind of crazy with my pen today. Okay. So let's look at 2 traits. Body color and wing shape. And I'm not going to use the positive versus no positive for wild type, because that gets kind of confusing. So I'll just do what Mendel did, okay? And what we do for Mendelian genetics, okay? So I'll say that G is body color, and this codes for a gray body, but little g codes for a black body. That is one gene, that is the G gene, right? Then we have capital letter L, codes for long wings. And then the lowercase l, which I'm not going to put a line over because it might confuse you as looking like an I, is codes for vestigial wings. Okay, so these genes are linked onto chromosomes. So if a fruit fly looked like this, had this genotype, right? This is the genotype. That is the combination of alleles, right? That's a capital letter L, okay? So they're heterozygous for both genes. What would they look like? Gray body with long wings, right? So they'd have a gray body and they'd have long wings. What is that called? Their phenotype. Okay, let's cross this with an individual that has this combination of alleles. So they are homozygous recessive for both, and so they would have a black body and vestigial wings, right? So they would be black body and vestigial wings. Okay. So we have to think about the way that these genes are grouped onto chromosomes. So if they're on the same chromosome, so I'm going to draw my chromosome, and I'm going to put my centromere there, and I'm going to say that this one, G is here, and L is here. This one, little g and little l, okay? So they're on the same chromosome. This one, both chromosomes are identical because they would be G, L, G, and L. Okay, so that means they're linked. Okay, so if we did a Punnett square, right, where we just combined the possible examples, so this could be like, if this was the male, this could be the sperm, right? So I could put my little sperm, and these could be the eggs. So that could be the female. It doesn't matter because this is not sex linked. Okay. So what are the combinations that we could get in the offspring? So this one could combine with this one, right? So I could get G big L, no, sorry, G big L, no, I have to put the G's together. There we go. I could get that combination. Okay, so I keep the G's and the L's together. So notice if I put these two together, I get that combination. If I put these two together, I would still get that combination. So I'm only going to write it once. And then notice I could get this, right? So this one could combine with one of these and give me this, okay? So the interesting thing about this is that these two offspring are exactly like the original parents, right? They are either gray body, long wing, black body, vestigial wing. So there's no variation. There's no like intermediate, like there's no like gray body, but vestigial wings, or black body and normal wings. Does everybody see that? Okay. So these are what are called the parental phenotypes. So these are called parental phenotypes. Okay. Did I go too fast on that one? Okay, put it back. Needed to go to my whiteboard, so I ran out of space. Okay, those are my parental phenotypes. Okay, so what Morgan found is that if we were doing this, what Mendel predicted, let's do what Mendel predicted first. Okay. So Mendel would predict okay, Mendelian predictions. If I have this combination, okay, then there would be an equal chance of getting a big G and a big L. So I could have big G, big L as being one of my sperm. I could have big G, little l, right, as being another sperm. I could have little g, big L, and little g, little l. So those are all the possible combinations that I could possibly get if they're not linked, right? So Mendelian predictions, they would not be linked. And he predicted that genes assort independently of one another. Okay, so that's sometimes called independent assortment. Okay, so that means that it doesn't matter. They would all get an equal probability of coming together in the gametes, right? And so this one's really easy. I could put GL. Okay, that's the only possible egg that the female could produce. So I don't have to write that 4 times. So I can kind of condense my Punnett square into a 4x1. Okay, so these come together in my Punnett square. And I always put the G's together and I always put the L's together. Okay. So when when Morgan was doing his work, this was what he predicted he would get. Okay. So 1/4 would be like one parent, and 1/4 or quarter would be like the other parent, and then you'd have intermediate. And so I'm going to call these recombinant offspring. Because you have a recombining, right? So this actually produces more variation, right? Because some offspring could be gray like one parent and vestigial wings like the other, or black bodied with long wings. You have more variation, okay? So if I had 1,000 offspring from this cross, right, I would predict 250 would be this, 250 would be that, 250 would be that one, and 250 would be that one. So there'd be an equal probability, right? So half of them would be like the parents, the other half would be recombinant. Okay? So these were the predictions that Morgan was working with. And what he found was more like, if you have 1,000 offspring, okay, so this is Morgan's results. Okay, if you have 1,000 offspring, 420 are this, 80 on average are this one, 80, and then 420. Okay. So we see that the Morgan's results were dramatically different than what you would predict if the genes were assorting independently. Does that make sense to everybody? Sort of? Okay. So, but if you think about the way that the chromosomes are on the, where the genes are on the chromosomes, you shouldn't get any of this, right? So why do we even get recombinant offspring? What mechanism can explain this 160 individuals out of 1,000? What can explain that? What mechanism happens in prophase 1 of meiosis 1? Anybody remember? Prophase 1 of meiosis 1, you get a shuffling of the chromosomes, and that is called crossing over, right? So this is explained by crossing over. Oh, shoot. There we go. Explained by crossing over. So remember, crossing over is the exchange of the genetic material between chromosomes. Okay? So that you're
Happening during meiosis.
prophase 1 of meiosis 1.
I'm a little concerned and confused where the data comes from.
Okay, so these are his results. This is what he would have predicted. So if he did that breeding and he came up with 1,000 offspring, on average, he got 420 that, 420 that, and 88. So this is just his results.
That's what we would predict if we were doing a Mendelian count. That's what was.
Yeah, so you would not have predicted this. There's no way that you could have predicted this. This is different. This is easy to predict because you get your Punnett score to predict it, right? So Morgan was like, whoa, something is happening. Why aren't these genes assorting independently? Why am I getting this skewed ratio, okay? And so then he started looking and he noticed that there were sets of traits that always had the skewed ratio. And then a light went on. He said, traits are inherited in groups. Genes are linked together onto chromosomes. And if they're found on the same chromosome, they're less likely to assort independently from one another. Right? So it's like this huge, big light bulb went on. It's like, you know, at that point they didn't know what a gene was. They didn't even know what the genetic DNA was, the genetic material. But they just knew that genes were being inherited differently in some cases, and so therefore they were linked. Okay? So let us go back to our picture of the chromosomes. Okay? So this is my picture of the chromosomes right here, right? So in order for me to get recombinant offspring, these, in just this parent alone, it happens in this parent too, but it doesn't matter. But in this one, right, the big L gets put over here and the little L gets here, and then all of a sudden we have G with big G with a little L, right? and little g with a big L, and that is what gives us the recombinant offspring. So the crossing over is what produces those gametes, and it doesn't occur very frequently compared to it not crossing over and separating. Okay? So this was the very first way they mapped genes onto chromosomes. So in your notes, you'll say that this is the first, looking at recombinant offspring is the first way they mapped genes onto chromosomes. Okay. Now you don't need to know this person's name. Actually, I don't ever test over names, but it was kind of interesting. Working in Morgan's lab was an undergraduate student. And he had this brilliant idea to map the genes based upon the percent of recombinant offspring produced. And so they call these, when you map them to chromosomes, they call them map units. So I'll say that 1% recombinant offspring. So if I got 1% recombinant offspring, then that would mean that my two genes, I ran out of room here, my two genes, so this is offspring, my two genes are 1 map unit apart. So this equals 1 map unit. Okay, it's like, oh. So what that means is that if the genes are further apart, then you're going to get more recombinant offspring. If the genes are really close together, you're less likely to get recombinant offspring. So if we look at our chromosome, right? If my G and my L are far apart, right? The possibility of them being separated by crossing over increases, and I get more recombinant offspring. If my chromosomes with the G and the L right on top of each other, remember crossing over is a random event, right? So if crossing over has to occur right there for the genes to be separated, then I'm going to get less recombinant offspring, right? So if you go back to my example, it was 80 plus 80, right? So if you look at the numbers, 80 plus 80 is 160, right, out of 1,000. And that is 16%. So 160 is 16% of 1,000. And so then what you would say is that G and L are actually (16) map units apart.
So if they're far apart, they're more recombinant or less?
If they are far apart, you'll get more recombinant offspring because there's lots of places where crossing over to occur. So they're likely to be separated during crossing over. If they're really close together, they're unlikely. So let's say like, if I got like, instead of 160, let's say I got maybe 200 total, that would be 20 map units, right? So I would have more, if I had 20% of my offspring were recombinant, I'd have 20 map units. What if I had only like 10? That would be like 1%. And so then I would only have 1% of my G and L would be like 1 map unit apart, and then only 1% of my offspring are expected to be recombinant.
So on the one where they're closer together, is there always the crossing over though, or sometimes?
Sometimes it doesn't cross over at all, and you wouldn't get any separation.
How do you know?
You would do the experiment. So you would do an experiment like Morgan, right? You would take your fruit flies and you cross them and you look at how many recombinant offspring you got, and you'd say, oh, well now I know the distance that those two genes are on the chromosome, because that's the number of recombinant offspring. So it's kind of this brilliant idea that this undergraduate student had, and it carries over today in genetics research. Okay.
So if you did the experiment over and over, you're gonna
On
see
average, yes. So you're going to see some variation, but on average, you would see that relationship. So that's the idea of linkage, of linked genes onto chromosomes. Okay, so the last thing we'll talk about is actually something that we've already talked a little bit about. And this is idea of chromosome number. So if you have the normal number of chromosomes, you are said to be a euploid. Right? So U means good. You've seen that in the word eukaryotic, a good nucleus. Ploid means number of copies, right? So in humans, right, this would be 46 total chromosomes, not pairs, but total chromosomes. Right? Now, when you put an A in front of a word, it means without. So this is called an aneuploid. And I can't put an A and have it be EU, so I put an N in there too, right? So this is without a good number. So this would be without the correct number. Now, we don't have a lot of examples of aneuploidy in humans, but like in other organisms, it can be very common, specifically plants. Plants are really weird in that they could actually be triploid. So they could be like, they could have 3 copies of every chromosome and be normal. They actually can survive all that extra genetic information. But in most cases, animals can't. And there's not a lot of examples of aneuploidy in humans. But we have what is called trisomy. So that means that you have 3 copies of one chromosome. And sometimes this is written 2n plus 1. So 2n means that you would have 2 copies of every chromosome, so that would be 46, right? But if you add 1, that gives you 47, right? So you have 47 total chromosomes, right? So we talked about Down syndrome as being trisomy of the 21st chromosome, right? So trisomy 21, right, is Down syndrome. Okay. We also have some examples of what is called monosomy. So instead of 2 copies, we have a single, oops, that should be monosomy. Write that again. So this would be 2n minus 1. And in humans, we have only one example of monosomy, and that is the sex chromosome. So the only example is XO, right? So the O is the missing sex chromosome. Right? So would you predict this individual, if they're XO, would they be male or female? Would they look male or female? Female, right? Because it's the Y chromosome that determines the sex. And so this would be a female. Right, so this is a female, but there are some characteristics that are associated with this, and it is called Turner's syndrome. So that's the only monosomy that we can survive with. You can't survive one 21st chromosome. You can't survive one second chromosome, right? You can only survive losing the second X chromosome. And this is probably because males only have one X chromosome. And then we're going to talk about what happens to the other X chromosome in a normal female, right? It gets deactivated, and we actually had mentioned it previously because it becomes the far body, right? Okay. So the process of getting an abnormal number of chromosomes is called non-disjunction. So this is the mechanism. So disjunction refers to the separation of the chromosomes. So we'll say that the chromosomes do not separate properly during meiosis. So in your book, there is an image of non-disjunction that looks like this. Okay. So notice that in meiosis 1 here, the chromosomes do not separate, right, properly. And so you actually get a gamete that is produced that is missing one chromosome. Right? So this is just a simplified example of just 2 pairs of chromosomes, right? You can also see that you could get an extra chromosome, right? Or you could be missing a chromosome. Now, most of the time, this is lethal, right? So most of the time, this is lethal, this non-disjunction, right? So if, for example, if you got an extra 2nd or 4th or 6th chromosome, for example, you would not be able to survive that. So the embryo would be inviable. So it might not even be able to develop enough to implant, or once it implants, maybe it doesn't produce enough hormone to be maintained, and so it's spontaneously aborted. So if you had an extra 2nd chromosome, or 4th chromosome, or 6th chromosome, or 8th chromosome, or any of the other ones, except for the 21st one, so for some reason, that 21st chromosome, probably because it's really tiny, right, that for some reason does not cause an inviable offspring. So people are able to survive that. Okay, so we have lab in a little bit. So make sure that you come to lab. And if you have any questions, let me know. Oh, yes, I need to sign up.